So welcome everyone to the microneurography webinar. My name is Nick. I am just your host today. I am joined by my colleagues Amy Chi and Pam Kacha. And just want to give you some housekeeping rules. So yes, you can ask questions in the chat or the Q&A. And if it's something trivial, then our team will answer these. Otherwise, please wait to the end and I'll just read out the questions and then Vaughan will be happy to answer. So without further ado, I'd just like to introduce our guest speaker here, Professor Vaughan Macefield, who will be doing a webinar for us on microneurography. So I'll just stop sharing my screen there. You can take it away. Thanks, Vaughan.
Thank you. Welcome, everybody. I believe there are some friends of mine around the world listening in, so hi. I'd like to see you in person again very, very soon. So I'm going to be just giving an overview on microneurography, which is a means of recording from peripheral nerves in humans. Just a brief overview. It was developed by Karl-Erik Hagbarth and Åke Wahlberg in Sweden in the mid-60s. I have the great pleasure to have worked with both of those and with their students. And I've trained many students myself. But as you can see, it's contributed a lot of information on tactile systems, proprioception, pain, sensory motor control, and control of the sympathetic nervous system. And I'll be touching on quite a few of these today. So tungsten microelectrodes can be inserted into any accessible peripheral nerve, such as the median or ulnar nerve of the wrist, or the upper arm, the perineal or tibial nerves at the knee, or the sural nerve at the ankle. And I'll also be talking about another approach we've developed. They can also be inserted into branches of cranial nerves, such as the inferior alveolar, supraorbital branches of the trigeminal nerve. And again, I'll be talking about another approach we've developed for obtaining information from another cranial nerve, the vagus nerve. So I use the NeuroAmp-X, the amplifier developed by AD Instruments. This was the, I can say that I was the design consultant for this. So I provided all the specs and tested all the prototypes, and it's a fantastic, dare I say, amplifier. And as you can see from the symbol here, it's safe for human connection. Now, AD Instruments put all of their hardware through a rigorous testing protocol to obtain this certification, and many other producers, such as WPI, do not. Even though their amplifiers are used in human situations, they always have a warning saying not for human use. This is not the case with the NeuroAmp-X. So it's got a very wide band pass, very high signal to noise ratio. And because we're recording from very small nerve signals, they had to be amplified 20,000 times. There is this head stage, which is connected to the front end, the NeuroAmp-X. This is made from high-grade stainless steel, and because of the shape of the amplifier, it is ideal for animal use too, because it can go into a Narashigi micro manipulator, so it can be inserted into the brain of awake animals, for instance. But I'm going to be talking about its use for human studies. So this is what we do. There are 3 terminals, as you can see here, and we attach a reference electrode, an active electrode, and a ground electrode, which is just a surface electrode attached to the skin. It's a differential amplifier, which means that it's measuring the voltage or the potential difference between the active electrode, which is inserted into the nerve, and the reference electrode, which is inserted just under the skin. And the reference electrode is basically a tungsten microelectrode from which a millimeter or 2 of the insulation has been removed from the tip. The active electrode is a high impedance microelectrode. And for single unit recordings, we typically want an impedance above 700 kilohertz. But often we can get very good recordings with lower impedances. So we use electrical stimulation to guide the microelectrode through the tip. We give weak pulses of 10 microamps to 1,000 microamps, so 1 milliamp, 200 microseconds in duration. And we use this to guide the microelectrode into a fascicle of the nerve. Now, for instance, the common perineal nerve at the fibular head is composed of distinct fascicles supplying either muscle or skin. And we know from experience that once we get a twitch or a radiating paresthesia, pins and needles, in the innervation territory at 20 microamps, we know we have entered the fascicle. And the advantage of this approach is that you can identify the fascicle based on the responses to intraneural stimulation. So if we're getting dorsiflexion of the ankle, we know that we've got tibialis anterior, we can see the muscle contracting. If we've got extensor hallucis longus, then just the big toe goes up, and so on and so forth. Now, often as we are recording, as soon as we penetrate the fascicle or move from one fascicle to another, we get this characteristic burst of impulses, which we call an insertion discharge. So a very high frequency discharge, and this is simply due to mechanical stimulation of the axons close to the tip of the microelectrode, generating ectopic impulses. And it's always wise to wait for that to settle down, because often it reveals that you've impaled a single axon, such as in this case, a muscle spindle afferent. This is a recording from a muscle fascicle, the common perineal nerve. Now, within the nerve, as I mentioned, it's composed of distinct fascicles supplying either muscle or skin. The only difference there for the common perineal nerve is extensor hallucis longus, the muscle that raises the big toe. There's also a little patch of skin just between the big toe and the second toe. So it's very important to be aware of that, which is particularly relevant when discussing sympathetic nerve recordings. You need to know that the recording is supplying muscle or skin. So here we have a microelectrode that's impaled a myelinated axon, recording from a muscle spindle ending. These are often spontaneously active. And down here we have a recording from a single muscle vasoconstrictor neuron. I'll come back to these later. But first I'll talk about the myelinated axons. And as you can see, they generate positive going spikes, whereas the unmyelinated axons generate negative going spikes. And that's just because unmyelinated axons, C fibers, do not have, by definition, the insulating myelin, so the action potential, the ion fluxes are occurring across the whole length of the axon rather than just at the nodes of Ranvier in a myelinated axon. So positive going spikes, myelinated axons, negative going spikes, C fibers or unmyelinated axons. So I'll first talk about myelinated sensory axons, afferents. This is a typical recording from a muscle spindle ending. As I mentioned, they're often spontaneously active at rest. This is in extensor hallucis longus, a muscle spindle primary ending. And just applying, we've got a force transducer over the big toe, and pushing back down on the big toe, represented by this negative going deflection, which indicates plant deflection. Pushing down on the big toe generates an increase in firing rate. And this is, of course, rate dependent. So slow push down causes a slow increase in firing rate. And you can see here different rates and amplitudes of muscle stretch. These are exquisitely sensitive stretch receptors located in muscle, and they are essentially our primary proprioceptors that tell our brain where our limbs are in space. Of course, cutaneous afferents contribute to that function as well, and I'll come back to that later. Muscle spindles are also responsive to active contractions, so voluntary contractions of the muscle. And you can see here that typically when we make a weak voluntary contraction, this is now an upgoing contraction, so a voluntary contraction lifting up the big toe. Here's the EMG and the root mean squared EMG, the smooth profile. The muscle spindle, because it's located in parallel to the muscle fibers, extrafusal muscle fibers, is unloaded during a contraction. But if the contraction is strong enough and you engage gamma motor neurons, the fused motor neurons, then that unloading can be offset and we can maintain this firing of the muscle spindle. So this is an important feature of muscle spindles. They're the only mechanoreceptors in the somatosensory system that have their own innervation, motor innervation in the form of gamma motor neurons, which can control the sensitivity of the muscle spindle to stretch. And muscle spindles, being very sensitive length detectors, they're particularly sensitive to very small length changes, such as occur during vibration. So if we apply a vibrating stimulus over the muscle belly, this is the same recording of the extensor hallucis longus, we can see that we can get entrainment or partial entrainment of the muscle spindle to the vibratory cycles. And here, even just applying vibration over the toenail, this is very, very remote from the actual receptor, which is located very high up in the shin. Exquisitely sensitive. So that mechanical stimulation of the toe, just applying a very small amplitude vibration, is transmitted up through the tendon, the extensor hallucis longus tendon, up to the receptor location. So it's very important, and I know many of you are just interested in the sympathetic nervous system, but it is important to know what you are also recording, which can help define whether you're in a muscle fascicle or a cutaneous fascicle. And also there's a wealth of information out there on the somatosensory system, there for exploring. So half of my work involves the sympathetic nervous system, the other half the somatosensory system. We recently further characterized the behavior of muscle spindles by using ultrasound to measure the changes in muscle fascicle length. And you can see here, this is the ankle angle. This is a fascicle length, muscle fascicle. So when I say muscle fascicle in this context, it is the fascicle of the muscle itself, not of the nerve. So changes in muscle fascicle length and velocity. This is a recording from a muscle spindle ending in tibialis anterior. And we're just applying very, very small amplitude sinusoidal rotations about the ankle. And we can see, as I showed you in that earlier slide, that firing rates are encoding both fascicle length and fascicle velocity. Now, these small angle changes are similar to those experienced during standing. These recordings were obtained in seated participants. We're recording from the common peroneal nerve, but we were interested in recording from muscle spindles in the foot, the tiny muscles in the intrinsic muscles of the foot during free standing, free unsupported standing. Now, this hadn't been done before. So we developed an approach where we inserted a microelectrode into the posterior tibial nerve, which is located behind the medial malleolus of the ankle. So here we have the head stage again, microelectrode inserted parallel to the Achilles tendon and directed towards the posterior tibial nerve. Now, this is the nerve, a branch of the serial nerve, which runs down behind the calf muscles and supplies the small muscles of the foot, many, many muscles in the foot, and also the skin on the sole of the foot. So we could record from both muscle spindles and cutaneous afferents in the sole of the foot during free standing. This hadn't been done before, and as I said, we had to develop this approach. The advantage of this approach is that there's when you stand up, there are very small changes in ankle angle if the leg is just supported, you know, seated and not resting on anything. But as soon as we stand, now this is the force from the force platform, this is the EMG from tibialis anterior and from the soleus muscles. You can see that a muscle spindle here, in one of the intrinsic muscles of the foot, responds to that force, the whole body weight being applied to the foot. Now, mostly these muscle spindles are silent at rest. We've characterized their behavior in seated conditions without any load, but importantly during standing. And you can see that this is a muscle spindle ending in the flexor digitorum brevis, a very small muscle that flexes the toes. And this, we've asked the participant to just push down on to actively contract the toes, push down on the force plate, and here, the subject is standing, so this is the background force, and you can see the force goes down as pushing occurs. And this is just spontaneous changes in firing of the muscle spindle during postural sway. And we've imposed displacements of the body on this motorized platform, so we can characterize how muscle spindles respond during free standing, which is exciting, we think. Now, cutaneous receptors, a very important class of mechanoreceptors. They come in different flavors, rapidly adapting, slowly adapting, and their firing patterns are really determined by the specializations of the mechanoreceptor ending. So they're all myelinated axons, but as you can see here, they have different features, different specializations of the actual sensory ending. And it's those features that determines the firing properties of these endings. If we look at the pacinian corpuscle, this is the axon, this is the generator regions, this is the part of the axon, the very tip of the axon, which has no myelin. But you can see that it's surrounded by these lamellae, fluid-filled layers, which essentially act as a high-pass filter. These have exquisite sensitivity to mechanical stimulation. They're the receptors in the skin that have the lowest mechanical thresholds. And because of this high-pass filtering feature, they only respond to very brisk mechanical events, such as blowing over the skin. These are the receptors that would respond to an ant crawling over your skin, but they respond to just blowing over the receptive field and even tapping remotely to the receptive field. They have very large fields, receptive fields, but as you can see, exquisite mechanical sensitivity. Another class of afferent, slowly adapting type 2 endings, which have large receptive fields, except there's a group that are located in the borders of the nails. Now they are slowly adapting, as you can see here. You apply an indentation directly to the receptive field. They have these well-defined hot spots, and they can generate high firing rates. But if we apply forces to the finger pad in different directions, you can see that they have a very marked directional sensitivity. Each ending has a preferred direction, which we can see here, different directions being applied and different amplitudes. So in here, we're applying a force at 10 degrees to the normal, 20 degrees, then 30 degrees over in C. And you can see that this ending, and indeed all the endings around the nail beds, have exquisite directional sensitivity. This makes them ideal for encoding contact forces applied to the finger pad. Okay, so now on to recording sympathetic nerve activity. I'll start with muscle sympathetic nerve activity. And So this is a recording with a fairly high impedance electrode. It's what we call an oligo-unitary recording. Oligo means few. So there are a few units in there. These are the negative going spikes, the action potentials generated from a few axons that are located close to the tip of the microelectrode. Now you can see the background noise of this recording is very low. This is, of course, recorded using the neuroamp. The background, the root mean square signal, very low, below, typically below 2 microvolts. And this means that we've got a very high signal to noise ratio. Now, that is a good recording, but not all recordings are as good as that. We can have a typical multi-unit recording. The background noise is quite high. There's a bit of ongoing afferent activity, but we can clearly identify these negative going spikes, bursts of muscle sympathetic nerve activity that are occurring spontaneously and have clear cardiac rhythmicity. This is the root mean square process signal, which is calculated in a lab chart. And this is the integrated nerve signal, which most people are used to. I prefer the root mean square because it gives a much smoother profile and it doesn't suffer from the time lag that an integrator introduces. So an integrator will introduce, the time constant is 100 milliseconds, it will introduce a time lag of 100 milliseconds, which is not of much consequence, but the root mean square being a moving average, there is no time lag. Now this is a typical multi-unit recording. On the right is an oligo-unitary recording, so just a few spikes firing. There's also these low amplitude spikes in the background. But when you have such a focal recording like this, you don't really see much in either the root mean square signal or the integrated signal. This is important to be aware of this because most people are just interested in this signal. They're not interested in the raw nerve signal in terms of they don't analyze it because most people are just interested in counting the number of bursts per minute, number of bursts per 100 heartbeats, measuring the amplitude of the burst, etc. But that is difficult when you have a recording like this. You would look at that, if you're just looking at these top 2 traces, you say, this is not a very good recording. But clearly you can identify sympathetic spikes in there and you can analyze that signal. And this is what we do. So, and it's also important to note when artifacts appear. So in this box, I've just highlighted in the RMS process signal. So again, this is we use a 200 millisecond moving average, and you can see that this is straddling this spike, this large electrical artifact. And we can clearly see that an artifact is defined as such because of its square shape, this rectangular shape. It doesn't have the smooth profile of a burst of sympathetic activity. So very important to be on the lookout for artifacts and to exclude them. Now, of course, most of you who are, well, all of you who are doing these recordings are very, very familiar with this. Now, we can use a lab chart, cyclic measurements feature in lab chart to detect bursts of sympathetic nerve activity. So here I've just set up within lab chart, within the cyclic measurements feature, in another channel, using the RMS nerve, the RMS process nerve signal as the input or the source channel, and we set it up so it detects a burst every time a certain threshold is reached. Actually, we use the, there are various presets. So if you're wanting to detect R waves, you can use the ECG preset. If you want to detect skin blood flow from the pulse plethysmograph, you can use that finger cardiovascular pulse preset. Actually, if you use the respiratory airflow preset, that works really, really well for detecting bursts of muscle-sympathetic nerve activity. Why? Because like airflow, these are slow events, relatively slow events, not like the R wave of an ECG. RMS processed nerve signals, bursts of sympathetic activity, are slow. So give it a go. It works very well for detecting bursts of muscle sympathetic nerve activity, which you can see here. It's done a pretty damn fine job of detecting these bursts. Peak parameters is a great feature once you've got it. So you can either detect bursts with cyclic measurements, as I've said, and just count the number of bursts in 100 heartbeats. You can count the number of heartbeats, of course. Or we would mostly use peak parameters to go through the file, jumping from R wave to R wave, and we use the time shift feature in LabChart to move the burst back in time so that they straddle the relevant R wave. So we, in this case, would move it back, you know, minus 1.25 milliseconds, depending on the size of the participant. Just so that we can bring the burst of activity close to the relevant R wave. And then using, we can write a macro to jump from R wave to R wave and use peak parameters just to measure bursts. So here there's no burst, so you get very small amplitude signals. But here we've got a well-defined burst, and we can measure the amplitude, the area, the width, rise time, fall time, anything that we like. And we can add that to the data pad. We can set that up to just automatically add to the data pad. We can use the spike histogram feature, which is very, very nice to detect negative going spikes of muscle sympathetic nerve activity. So here we've got the spikes, the sympathetic spikes. Down below, we're using the same program to detect the ECG events, the same program to detect the peaks of respiration down here. And then we can generate cross-correlation and autocorrelation histograms. So here we have the autocorrelation histogram of the ECG. So time 0 is the first R wave. Here's the times of occurrence of the subsequent R waves and the R waves preceding. So this is analyzing the whole file. We've got like 14,000, 15,000 events here. And you can clearly see in the cross-correlation histogram above that we've got marked cardiac modulation. We can quantify this. We can also do this with respect to respiration. And using this approach, we've shown that although skin sympathetic nerve activity is typically considered not to have cardiac modulation, if you use this approach, you can actually identify cardiac modulation. Importantly though, extracting the negative going sympathetic spikes, which are very, very narrow, can be used to get a clean recording from a noisy recording. So here, the subject tensed up. So we've got some EMG. Now, if we look at the spike histogram profile, this is the cluster of sympathetic spikes. It's not a single unit recording, of course, so we've got quite a cluster. But the wider EMG spikes, this is time along here, are shown in this cluster. So using spike histogram, we can just extract the negative going spikes from the EMG shown here, so the sympathetic spikes, and we can reconstruct the RMS process signal just from these extracted sympathetic spikes. This is the root mean squared process of the whole nerve signal. And you can clearly see that before the EMG kicks in, we can identify clear bursts, but when the EMG kicks in, there's a baseline shift and a lot of these bursts are hidden. Now, this is a very weak contraction that the subject just happened to be making. It's just one big motor unit there. But often, of course, there's a lot of EMG and the signal is right royally swamped So you can use this approach to extract the sympathetic spikes. We have used this successfully to record from sympathetic nerve activity to contracting muscles, and shown that as contraction intensity of the muscle from which we're recording sympathetic nerve activity increases, we can see a linear increase in muscle sympathetic nerve activity represented as spikes per minute. So again, these are the extracted sympathetic spikes, and we can see that up to, we've performed contraction intensities of 50% maximal. So very, very strong contractions. If you looked at the raw nerve signal, it's totally swamped by EMG. However, you can go in there into the raw nerve signal, and using spike histogram, you can extract the negative going sympathetic spikes from the positive going muscle spindle afferents, Golgi tendon organ afferents, and the broader spikes of EMG of the motor units. So a very powerful analytical tool. A lot of this is discussed in this recent review I wrote for Clinical Autonomic Research, which is freely available. Now I'll just talk about recording from single sympathetic axons. We use the same approach, but here we use a high impedance microelectrode. So we guide it into the muscle fascicle or a cutaneous fascicle, and we're just trying to find sites within the fascicle in which we can see a spike that stands out from the noise, clearly from the noise. And when we superimpose all these spikes, we can clearly see that the spike morphology is uniform. So here is a muscle vasoconstrictor neuron, a single sympathetic axon supplying blood vessels and muscle at rest during a maneuver, a maximal inspiratory effort. This is another recording where we're getting a maintained augmented outflow. So we get bursts pretty much with every heartbeat, but clearly this individual neuron is just firing away, and we can see superimposed spikes. From the superimposed spikes that we are recording from one and only one axon. The caveat there is with any single unit recordings, there could be two axons of identical spike amplitude and width, but the chances of that are very, very low, and the firing rates that we find do not support that interpretation. So when we count the number of spikes, if we count the number of spikes per burst, you can see that they're mostly firing only once. Here it's firing three times here, but mostly once. Here's an artifact which was ignored, thank goodness. Here, during this augmented outflow, the neuron is firing once per burst, despite these very, very large bursts. And we can characterize the firing patterns. Here, we're just showing in all the cardiac intervals how often a given unit discharges. Mostly, as shown by the white bars, they are silent. So mostly 0 spikes per cardiac interval. And that is true for muscle vasoconstrictor neurons, cutaneous vasoconstrictor neurons, and pseudomotor neurons supplying sweat glands. Firing probabilities are similar, around 30–35%. And if we just look at those cardiac intervals in which a neuron is firing one or more spikes, you can see that the firing patterns are remarkably similar. And I've done modeling to show that this firing pattern can largely be explained by these postganglionic sympathetic neurons. These are what we're recording, postganglionic neurons. Their firing properties can be explained by them being driven by, on average, 1 to 2 preganglionic neurons. And these same patterns, mostly firing once per burst, can be seen in different pathophysiological states. Heart failure, bronchiectasis, obstructive sleep apnea, chronic obstructive pulmonary disease, the mostly firing ones. But you can see here that in these three conditions, unlike heart failure, there is a shift towards multiple firing. So there are fewer solitary spikes and more double and triple spike events. But on the whole, individual neurons primarily fire once per burst. So it would appear that rate coding is not an important facet by which sympathetic outflow is graded. Kevin Shoemaker and his team have used other approaches to demonstrate that, in fact, recruitment of silent neurons is the primary means by which the sympathetic nervous system grades its output. So I'll talk now briefly about a recent, well, not so recent, 15 years ago, we developed the approach of, 12 years ago, of recording sympathetic nerve activity while scanning the brain. We've been imaging the brain during maneuvers such as a maximum respiratory breath hold, hand grip, post-exercise ischemia, etc., in the scanner, because, you know, these are standard maneuvers. You know that if you're going to take a deep breath hold or perform a Valsalva, you're going to get ongoing bursts of muscle sympathetic nerve activity. Likewise, if you do hand grip and post-exercise ischemia, you're going to get a sustained increase in activity. And we've been scanning the brain, trying to identify the nuclei within the brainstem and cortical areas in which we could, to identify areas in the brain responsible for the generation of these bursts. But we were interested in seeing whether we could do that at the same time as recording nerve activity, which hadn't been attempted, because we really wanted to see what is responsible for generating spontaneous bursts of nerve activity. So, yes, so now importantly, because the head stage of the neuroamp is made from high-grade stainless steel, is it intrinsically compatible in a magnetic resonance imaging environment? It's high-grade, military-grade stainless steel, in fact, so although it has iron in it, it can't be magnetized. Now, because the microelectrodes are tungsten and the wires connecting the microelectrode to the head stage are copper, the pins are gold, there is no issue there, and so we managed to record both muscle sympathetic nerve activity and skin sympathetic nerve activity in healthy individuals, as well as different pathophysiological states such as sleep apnea. We're currently looking at individuals with different forms of hypertension. But MSNA coupled fMRI and SSNA coupled fMRI, which is what we call it, allows us to identify regions in the brain responsible for their generation of the burst. So here's the approach. This is a 3T scanner. We've got the head stage there. The amplifier is in the control room. We're actually also doing this at the 7T facility in Melbourne, and I developed the background for this at the 7T facility in Nottingham. So all the physicists there measure the temperature of the electrodes in the scanner when they were embedded in agar. They were satisfied that there wasn't going to be any heating. And then we made the first recordings at 7T in Nottingham. But now Tyder Wood, my colleague here at the Baker, and I have been recording muscle sympathetic nerve activity and skin sympathetic nerve activity at 7T. That's ongoing work. But the very first recording we made in the 3T scanner, this was the scanner in Sydney. So we've got these scanning artifacts, and we can't see anything in the nerve signal during that. And we could hear over the loudspeaker where there's whoosh, whoosh, but it was really hard to see. You can sort of make out that there's a burst here. But then we did what good scientists do. We applied a filter, a high-pass digital filter. This is offline in LabChart. And we can see the same recording now with a high-pass filter, digital filter, we can clearly see these bursts. So back to the unfiltered, filtered, gets rid of all this background noise, scanner noise, and we can identify bursts. So basically, as soon as we save the recording, this filter kicks in and gets rid of all this background noise. So despite the high magnetic field, we can record sympathetic nerve activity from the common peroneal nerve while scanning the brain. We can measure the amplitudes just using peak parameters, as I spoke about before. And the approach we use is a sparse sampling method, where for about half an hour, we are scanning 4 seconds on, 4 seconds off, 4 seconds on. So loud scanning artifacts here, and then we hear the whoosh, and we can record the burst of activity in the off period. And because of the nature of blood oxygen level dependent signal, which is what we're recording in the brain, there's a 5-second lag from electrical events in the brain and the appearance of the bald signal. So that's a 5-second delay. There's also a conduction delay from the brain at the sympathetic volley that we're recording at the level of the knee. It takes about a second to travel down. So 5 minus 1 is 4 seconds. So that's why we use this 4 seconds on, 4 seconds off, and we measure every second within this 4-second off period, the bursts. So here we've got a burst and we're looking ahead in time, 4 seconds later, see what's happening in the brain. So this burst that was recorded in the periphery, we're looking 4 seconds ahead and seeing what lights up in the brain. What lights up is rostroventral medulla, increase in signal intensity, warm colors, decrease cool colors, so decrease in caudal ventrolateral medulla, nucleus tractus solitarius. We've identified the caudal pressor area. So these are the same areas that we know operate in animals, experimental animals. All this basic baroreflex circuitry that is identified in the rat and the cat, etc., is present in humans. Bear in mind that the human medulla is basically the size of the tip of my pinky. So we're really pushing the boundaries. We can also identify areas in the cerebellum, ventromedial and dorsomedial hypothalamus, the insula, and dorsolateral prefrontal cortex, interestingly. So this is an area that people hadn't really thought contributes to control of blood pressure, but clearly it does. And in fact, we're using this knowledge. Ty and I have been, and our students have been running a series of experiments where we're applying weak sinusoidal currents to the dorsolateral prefrontal cortex, and we can see that it causes modulation of sympathetic outflow to both muscle and skin, which is unpublished, but also very, very exciting. So we can look, having identified all these areas in the brain, we can then go on and look at how they're connected to each other. So we use what's called functional connectivity analysis. So here we're looking at the ventromedial hypothalamus, which is very strongly coupled to the generation of muscle sympathetic nerve activity. We basically put seeds here. So it's basically doing a multiple linear regression between this and all these other areas. And we just plant the seed there and we can see what is functionally coupled. What is functionally coupled with the ventromedial hypothalamus is, importantly, rostral ventrolateral medulla. You saw that in the brainstem. That is a primary output nucleus of muscle sympathetic nerve activity. And we know that from animal work, but also our own human work. So this is strongly coupled to the hypothalamus, as is the anterior insula, just on the left, curiously, the dorsolateral prefrontal cortex on the right, and also, curiously, precuneus. And this is one of the most metabolically active areas in the brain. Its activity is high when you're awake, it goes down during sleep. And we also know that muscle sympathetic nerve activity goes down during sleep, deep sleep, as does blood pressure. So we're thinking that this area here, and perhaps also dorsolateral prefrontal cortex, contribute what, for want of a better term, a wakefulness drive to sympathetic outflow to the muscle vascular bed. When we're awake, our MSNA is higher than when we're asleep. And we think it's because of this higher cortical region. Again, we can look at connectivity by looking at the seed, looking at the rostroventral medulla. We can see that if we look at that area, we can see that it is coupled to ventromedial hypothalamus, as we showed before, insular, precuneus, and also posterior cingulate cortex. And also we can look within the brainstem. So it's also coupled to the dorsolateral pons, the midbrain periaqueductal gray. Ventromedial hypothalamus is here, and dorsal medial hypothalamus. So very, very powerful approaches, analytical approaches, which basically depends on the feasibility, really depends on using this MR compatible amplifier, the NeuroAmp X. So finally, in the last 10 minutes, I'm just going to be talking about an exciting piece of work we started last year. We managed to get a few recordings before the big lockdown, COVID lockdown that happened in Melbourne and elsewhere, where we couldn't do any experiments. But again, this was with my colleague, Ty Dawood, and also Leah Wright here at the Baker Institute, an ultrasonographer, and a delightful colleague, postdoc from Italy, Matteo Viani, in which we used ultrasound to record from the vagus nerve. Now, a bit of background. We know a lot about how the sympathetic nervous system operates in humans, in health and various diseases, but we know very little about parasympathetic nervous system. Most of that knowledge is very, very indirect and is inferred, really. Inferences from measurements of heart rate variability. So nothing direct. And even the inferences that people make about the low frequency content of heart rate variability, reflecting sympathetic outflow to the heart, there is very little, essentially no evidence to support that. Work done by Murray Esler here at the Baker Institute many years ago, in which he measured noradrenaline spillover to the heart and tried to correlate that with low frequency heart rate variability, showed there's no correlation. More recent direct recordings of cardiac sympathetic outflow from Clive May's group at the Florey Institute here in Melbourne. Direct recordings of cardiac sympathetic nerve activity in the sheep. Again, no correlation to low frequency heart rate variability. Most people, on the other hand, would agree that the high frequency component, essentially respiratory sinus arrhythmia, reflects the cyclic withdrawal of vagal outflow to the heart. So the resting heart rate, as you're all aware, is kept in check by the vagus nerve. The intrinsic cardiac rhythm is much higher than the resting cardiac rhythm. So there's essentially negligible sympathetic output to the heart at rest anyway, and but there is ongoing vagal outflow, which is responsible for keeping heart rate low. And that cyclical withdrawal is responsible for the respiratory sinus arrhythmia we see. But until now, no one had recorded from the parasympathetic outflow in humans. So again, just back on the heart rate variability, it's grossly overinterpreted and is often applied as a general index of parasympathetic tone on the inference that this tone means that parasympathetic outflow to the heart is identical to parasympathetic outflow to the bronchi, is identical to parasympathetic outflow to the gut, which clearly is not the case. Anyway, earlier this year, as I mentioned, we conducted our first recordings from the cervical vagus nerve using ultrasound guidance. Now, people hadn't attempted this before because obviously the vagus nerve runs next to the carotid artery and the jugular vein, and it's kind of tiger country in there. So we used ultrasound guidance. So this was an approach developed popularized by Nisha Charkoudian and Tim Curry at the Mayo for going to the common perineal nerve in obese people. And it clearly works here. So this is a very handsome head here. And we've got the microelectrode being inserted. Here's the head stage, the amplifier, the active electrode being guided into the cervical vagus nerve. We've got the reference electrode here too. This is an image showing the ultrasound probe, which you can see here. And basically we can identify the common carotid artery, the internal jugular vein, scalene muscles, sternocleidomastoid muscles, and this baby, the vagus nerve. We can actually identify individual fascicles. We're looking to get this is with a standard 12 megahertz ultrasound probe. We're looking to purchase, if we get funding, a more powerful probe that would allow us to identify fascicles with much higher spatial resolution. Here we can see the electrode trajectory. And as we're approaching with live ultrasound imaging, we can see all these tissues being pushed out of the way, and we can actually impale the vagus nerve after pushing it a little. So we just have to push through the nerve sheath. We have managed to record spontaneous activity, which has respiratory related changes. This was showing ongoing modulation of discharge during breathing. We believe it was probably supplying one of the laryngeal adductors. We also, and this I should point out, here we were just holding the microelectrode. So we just got this recording when holding it. So there's some fluctuations in the amplitude. Nevertheless, with spike histogram, we could follow the spikes and plot the firing rate, the instantaneous firing rates. And we can see that the RMS signal increased with these changes in amplitude, but importantly, we can detect little bursts, which had a clear cardiac modulation. And this is shown here in a multi-unit recording. So we think that this recording, because it had this inverse relationship to heart rate and a positive relationship to cardiac interval, RR interval, you can see that they're clearly superimposed here, that this was a spontaneously active parasympathetic axon supplying the sinoatrial node. And this multi-unit recording, we believe, various reasons that I won't go into, that it was detecting multi-unit activity arising from the atria. So what are the applications of this? Well, as I've shown, we can record from cardiac preganglionic parasympathetic axons direct to the sinoatrial node and measure their firing rates and discharge variability the first time in humans. We can compare these to the known firing rates of sympathetic axons that we've been recording. We know that they mostly fire about up to 1 hertz, so 0.5 to 1 hertz. So sympathetic axons, postganglionic sympathetic axons have very, very low firing rates. Parasympathetic preganglionic axons, on the other hand, have higher firing rates, and they're myelinated axons. We can examine how these firing properties are affected in different disease states, different cardiac dysrhythmias. We can ask the question, does the vagus nerve play a role in limiting atrial fibrillation? We can record from baroreceptors in the aortic arch, see how their firing properties are affected by hypertension or connective disease disorders such as connective tissue disorders such as Marfan syndrome, which has an enlarged aorta. And by recording from the low pressure baroreceptors located in the atria, the contiguous veins, we can see how changes in posture affect their firing. So we're going to be resuming these experiments this year, now that we're back from lockdown. Having participants on the tilt table, get a recording, tilt them up and unload the low pressure baroreceptors. And we can look at how their properties change in POTS, for instance, postural tachycardia syndrome. And we can obviously see how the parasympathetic axons going to the heart are affected during changes in posture. And again, look at various disorders of postural hypotension. So it does open up an exciting new avenue of research. And yeah, we're really quite thrilled that people have shown a lot of interest in this approach. It's very early days yet. We've just got that first publication in JPhysio, but we're obtaining more data now, so watch this space. So look, thank you for your attention. Do feel free to contact me on this email. If you want to look at my papers, there's that ORCID ID if you want to make note, but yeah, happy to take any questions. Thank you very much.
Thank you very much, Vaughan. We do have some questions coming in now. I have one simple one. First, from Öztürk Khan. I do apologize if I mispronounced that. How can I generate RMS nerve from the raw MSNA signals?
Well, you could. Well, with the see, I use the Macintosh version of LabChart, which, in my opinion, is superior. It has a feature called RMS. So you find an empty channel, you identify the nerve as the source channel, and you select RMS, and you say, moving average and 200 milliseconds. Now, in the Windows version, you can also generate an RMS processed nerve signal just using arithmetic. And if you look on the website and just type in a search for how do I generate the RMS nerve signal on LabChart, it can show you, just use arithmetic, the arithmetic feature.
Great, thank you very much. Just waiting on some more questions now.
Pleasure.
Okay, we have one from Eric Dubois. When recording from human vagus, can you definitely identify A delta from C fibers? Also, I guess the system would work on afferent nerves, isn't it?
Yeah. So we can identify myelinated axons should have positive going spikes, but it's also important to note that even when recording from any peripheral nerves, you occasionally get negative going spikes from myelinated axons if, by chance, the tip of the microelectrode is close to a node of Ranvier. Then you see the spike is negative going, and that was one of the examples that I showed. So yes, we should be able to identify myelinated, so A delta axons and C fibers. C fibers will of course be negative going. So was there another part of that question? I'm not sure.
I think the last thing they said was also, I guess the system would work on afferent nerve fibers.
Yeah. Yep. So so we believe we were recording from atrial receptors. So yeah, we can record efferent and afferent activity from the vagus nerve.
Just a follow up there from Eric. Indeed, I guess it's in vitro, but can you assess the CV which allow to discriminate?
Sorry, CV coefficient of variation?
Let's see if they can expand on that.
Because I can't see the question.
It's just under chat, actually.
Oh, okay, yeah.
Yep.
That's great. Chat.
I meant that I can assess the conduction velocity in vitro.
Oh, beg your pardon. Yeah, conduction velocity. That would be difficult. You'd need, it'd be very difficult. You'd need 2 electrodes in the nerve. Theoretically possible, but wow, it's hard enough getting 1 electrode in the nerve. I guess you could use spike triggered averaging. If you know the distance to the atrium, say, estimate the distance, and knowing the time delay for the afferent recording, yeah, you should be able to work out the conduction velocity. Yeah.
Thank you, Vaughan.
That's the question.
Just waiting on some more. Eric has expanded here. I agree, very difficult in vivo. John Addock does it in vivo, but involves surgery to place the electrodes, so only used on animal models.
Yeah, and in fact, when I did my honors year, my first foray into research in the lab, I did direct recordings from the vagus nerve in the rabbit, so surgically exposed, cut the nerve and splitting individual nerve fibers in the paraffin-oil bath and putting them on hook electrodes. So yeah, very, very different approach, animal work versus humans. But of course, we can get very rich information from tungsten microelectrodes in humans. We can record from, as you've seen, single axons, both myelinated and unmyelinated, and get a level of detail that is often typically only seen in experimental animal work. But thank you for your comments.
Any more questions?
That's fine if there are no more, because we do have a participant about to come into the lab, and it never stops here. It's just ongoing research, pushing back the frontiers.
Fantastic. Okay, no problems at all. Obviously, everyone can contact Vaughan on that email address. If I can just quickly share my screen, that's fine, Vaughan. If anyone wants to contact us, I'm more than happy to do that at ADI. We do have a sales team here, and for any of your questions regarding the NeuroMap EX, but I'm sure Vaughan will have a much better understanding of that than we have. But thank you very much anyway. All the best, Vaughan. So we look forward to seeing everyone to our next webinar.
And thank you, Parimal, for organizing this, and Nick for your support, and thank you everyone for tuning in. And yes, I'm always happy to field any questions on the analyses that I've spoken about today. But yeah, again, if you look what's coming out, it is available online now, freely available online in Clinical Autonomic Research, a review article that I wrote on recording and quantifying sympathetic nerve activity in humans. So yeah, thank you for your attention, everyone.
Yeah, thank you very much. Okay, all the best. Thank you, everyone.
Bye-bye.